first off, with psychiatric disorders, they're very complicated in their genetics, and we don't really understand what causes these disorders. So what we're trying to do at the Haggerty Lab is find a common core pathophysiology that's in these disorders and eventually develop novel therapeutics for these disorders. And we're doing that by looking at the Wnt GSK3 signaling pathway. And the protein that I'm specifically interested in is DISC1, which stands for disrupted schizophrenia. And it's an important regulator in neurogenesis, and it inhibits GSK3, which is an enzyme that's targeted by many drugs for neuropsychiatric disorders. And it does this inhibition by physical interaction. So what we want to do is have a model to look at to try to better understand these disorders, and to do that, I'm more interested in the NPCs and the neurons. And to get the NPCs, we first started with patient skin cells, and then with those fibroblasts, we programmed them to induce pluripotent stem cells. And the nice thing about iPS cells is that you don't need an egg, whereas for embryonic parthenonuclear transplant you do. And back to the iPS cells, with those we can induce them to neural rosettes, and with the neural rosettes we can obtain NPCs by either hand-picking them or fat sorting them. So the first thing I learned to do was culture and passage NPCs, and here are a few images. So they had to be passaged about twice a week, and I would culture them Monday, and so this would be Tuesday, Wednesday, Thursday, and I just passage them and do that every week. After learning how to culture and passage NPCs, I then differentiated them, and here you can see as they differentiate through time, they become more and more sophisticated in their processes. Now here, I have 12 differentiated NPCs that are stained with pan-neuronal markers, which are just general dendritic markers. So I used MAT2, which are the dendritic markers, and SMI are the axonal. The blue dots that you see are just the nuclei. But as you can see in this image, it's very complicated, and there are a lot of neurons, and we can't really tell whether this axon is for that nuclei or whether it's for that nuclei. So what we want to do is we want to transfect individual neurons, or a few amount of them, with GFP. So in this image, you can see that there are a lot of neurons, but only one is stained here. Now, if we zoom in and look at the neuron a little bit closer, we can perform what's called a Shaw analysis, which is a way to quantify how sophisticated these neurons are in their morphology. So in a Shaw analysis, what you'll do is you'll have a center circle around the soma, and so for the first circle, you'll just count, well, for all the circles, you'll count how many intersections there are. And so for the first circle, you'll have 1, 2, 3 intersections, and you'll just keep going and going until you capture the whole neuron. And when you plot that, you'll get something similar to this, and this is the work of a postdoc in the Haggerty Lab. Here he had a control line, and he compared that to a cell line that was overexpressing a microRNA. And so you can see that the one that's overexpressing microRNA, it is a lot less sophisticated than the control. And I just want to mention that in this area, you tend to get dendritic intersections, whereas the further you go out, you get more axonal. Now, as mentioned earlier, DISC1 plays a role in neurogenesis, and what we're trying to do is dissect the role of DISC1. And so I'm working with 4 different cell lines. 2 of them are controls and 2 are DISC1 knockdowns. Keep in mind that I'm working with NPCs that have DISC1 knockdown in them, but they came from short hairpin RNA that silence the DISC1 gene. and we did that via RNA interference. So first, with NPCs, we looked at how they proliferated, and we didn't see much of a change, much of a difference in these cell lines. But when you differentiate them, there's a drastic difference between the two cell lines. So here are the two scramble controls, and here are the two DISC1 knockdowns. It's kind of hard to tell because there are a lot of dead cells, which are the white dots, but in population density-wise, the scramble control has a lot higher density. Now, if we transfect them with GFP, here are just examples of images that I took. So I took about 10 images of each cell line, and we're in the process of trying to quantify how complicated each cell line is. But as I was taking the pictures, I noticed that it was easier to find the scramble control neurons compared to the DISC1 knockdowns. So what we'd like to do is eventually use a microscope that'll take random images or non-biased images and then calculate an average of how sophisticated these neurons are. So when it comes to differentiating, there are two ways you can do that. You can do that by either growth factor withdrawal, or you can try an accelerated version, and we do that by inducing them with neurogen. And for the neurogen-induced method, we use astrocyte conditioned media. And I want to point out in this cartoon that in the brain, it's not just neurons. There are other types of cells in there. And here is a cartoon just showing how the astrocytes are like star-shaped glial cells in the central nervous system. And they're there for support above the neurons. And they can also release and uptake neurotransmitters as well. So here are the human astrocytes that I worked with and cultured so that I could make the conditioned media for the accelerated differentiation. For my project, I'd like to thank Wendy for providing guidance and assisting me with certain things. Jasmine for providing the lipopectomine and the DNA so I could transfect the neurons. Shailin for providing the astrocytes. Dr. Haggerty for letting me work in his lab. And Sue for allowing me and the